So, good afternoon, everyone. I am Darlene Guritan, and my report is all about Chapter 13, which is Intracellular Membrane Traffic, presented by me, Guritan Darlene Jyoti, from BSAD Science 3. So, objectives. Here is my semi-detailed lesson plan. At the end of the lesson, the students should be able to identify the molecular mechanisms of membrane transport, know the transport from the endoplasmic reticulum through the Golgi apparatus, transport from the Trans-Golgi network or TGN to lysosomes, transport into the cell from the plasma membrane or what we call endocytosis, and transport from the Trans-Golgi network TGN to the cell exterior or exocytosis. So topic, intracellular membrane traffic, time allotment 2:30 to 3:30 p.m. and here are my references as well as the materials that I use. Canva presentation, Google Meet, Google Classroom, CapCut, and Cellphone. So for the procedures, introductory activities, daily routine, we have the greetings. The presenter will greet the professor and the students. The presenter will also lead the prayer. As well as the checking of attendance, the presenter will ask someone to screenshot the attendance. So, priming, review or drill, icebreaker, jumbled letters. The teacher will know if the students have an idea about the topic by guessing the correct answer or word. So, that is the evaluation, a total of 20 items quiz and 10 points assignment. So now let's move on to the icebreaker before we proceed to the discussion, which is the jumbled letters. So, who wants to answer? Let's see if tama yung sagot mo. Yes, cell membrane. Next. Let's see if tama yung sagot mo. Next. Trans-Golgi network. Next. Lysosomes. Correct. Next. Exocytosis. And thus, endocytosis. So, thank you sa lahat ng mga nag-participate. Let's now proceed to our discussion. Key concepts, we have the molecular mechanisms of membrane transport, transport from ER to Golgi apparatus, transport from Trans-Golgi network to lysosomes, transport into the cell from the plasma membrane, which is endocytosis, and transport from the Trans-Golgi network to the cell exterior, which is exocytosis. So, introduction. The smallest unit of life is the cell. Cells are considered to be living because they display all of the characteristics of life. The characteristics of life includes metabolism, being made up of cells, growth and development, response to the environment, homeostasis, and the reproduction. All these things happen inside the cell. So every cell must eat, communicate with the world around it, and mabilis yung respond nila sa mga changes ng environment. So to help accomplish this task, cells continually adjust the composition of their plasma membrane and internal compartments in rapid response to need. So they use an elaborate internal membrane system to add and remove cell surface proteins such as yung mga receptors, ion channels, and transporters. Through the process of exocytosis, yung secretory pathway, nagde-deliver sila ng mga newly synthesized proteins, carbohydrates, saka mga lipids, either sa plasma membrane or sa extracellular space. By the converse process naman, which is yung endocytosis, ang nangyayari is yung mga cells, nagre-remove sila ng mga plasma membrane components and then dinedeliver nila to sa internal compartments, which is tinatawag natin ng mga endosomes, na kung saan pwede sila dong ma-recycle sa same or different regions ng plasma membrane or pwede silang ma-delivered sa lysosomes for degradation. So, yung mga cells, ginagamit din nila yung endocytosis para mag-capture ng mga nutrients such as yung mga vitamins, cholesterol, and iron. These are taken up together with macromolecules which is nagbabind sila and mamumove sila sa endosomes. Mamumove sila sa endosomes and then sa lysosomes. Na kung saan, dun sila pwedeng i-transport into cytosol for para magamit sila sa mga various biosynthetic processes. Pure space or tinatawag natin na lumen of each membrane enclosed compartment along the secretory and endocytic pathway is equivalent sa lumen ng mga other membrane enclosed compartment and then ng cell exterior. So, yung mga proteins, pwede sila mag-travel sa space na to, sa space na ito, which is yung interior space, kahit hindi na sila mag-cross sa membrane, which is nagpapas sila from one compartment to another. Dumadaan sila sa isang compartment to another compartment by means of numerous membrane-enclosed transport containers. So itong numerous membrane-enclosed transport containers, ang term na pwede natin gamitin dito is transport vesicle to apply to all forms of containers. So, within the eukaryotic cell, yung mga transport vesicles daw is tuloy-tuloy lang yung pagbud off sa isang membrane and then nagpo-fuse sila sa another membrane, which is dala-dala nila yung mga membrane components and soluble luminal molecules na tinatawag natin na cargo. So, itong vesicular traffic na to is once na nag-flow siya along sa mga highly organized directional routes, inaalaw nito yung cell na mag-secrete, na mag-eat, and then na mag-remodel sa sarili niyang plasma membranes and sa mga organelles. Form yung function ng mga transport vesicles. Each transport vesicle that buds from a compartment must be selective. It must take up only the appropriate molecules and must fuse only with the appropriate target membrane. So once na yung transport vesicle nagbud siya sa isang compartment, kailangan syempre yung transport vesicles is doon lang siya magpo-fuse sa appropriate target membrane, and hindi siya dapat mag-fuse kung hindi siya appropriate dun sa isang membrane na yun. So now, let's proceed sa the molecular mechanisms of membrane transport and the maintenance of compartmental diversity. Yung vesicle transport, nagbe-mediate siya ng continuous massive exchange of components between the 10 or more chemically distinct membrane enclosed compartments that collectively comprise the secretory and endocytic pathways. So may tanong dito na, with this massive exchange, how can each compartment maintain its special identity? So una, kailangan muna natin makonsider ano nga ba yung nagde-define sa character ng isang compartment. So it is the composition of enclosing membrane, which is yung mga molecular markers is naka-display sila sa cytosolic surface ng membrane, which is nagsuserve as guidance cues. Molecular markers na naka-display sa cytosolic surface ng isang membrane is nagsuserve sila as guidance cues for incoming traffic to ensure na yung mga transport vesicles is magpo-fuse lamang sa correct compartment. So next, how are these membrane markers kept at high concentration on one compartment and at low concentration on another? So dito naman, kailangan natin i-consider kung paano nga ba yung mga patches ng membrane is na-enrich or na-deplete sa mga specific membrane components, and then paano sila nagbabad off from one compartment and transfer to another. There are various types of coated vesicles. Mostly yung mga transport vesicles daw is nabubuo sila galing sa mga specialized coated regions ng membrane, which is nagbabad off sila as coated vesicles. So before the vesicles fuse with the target membrane, they discard their coat. So bago mag-fuse yung mga coated vesicles sa isang target membrane, nagdi-discard muna sila ng kanilang coat, which is required for the two cytosolic membrane surfaces to interact directly and fuse. So there are two main functions of coat that are reflected in a common two-layered structure, which is yung inner coat layer, nagko-concentrate siya sa mga specific membrane proteins sa specialized patch, which then gives rise to the vesicle membrane. While si outer coat layer naman is like a curved basket-like lattice na kung saan dinedeform nito yung membrane patch and sineshape nito yung vesicle. Three well-characterized types ng mga coated vesicles na distinguish by their major coat proteins. So, una, Clathrin-coated vesicles. So, yung nasa kaliwang picture is, ayan yung itsura ng Clathrin-coated vesicles. which is ang mediation transport from Golgi apparatus and from plasma membrane. It is the first coated vesicles to be identified, and then nagtatransport siya ng material galing sa plasma membrane and then between endosomal saka Golgi compartments. So, yung major protein component ng Clathrin-coated vesicles is yung mismong sarili niya, which is yung mismong Clathrin niya. So, which is nagpo-form yun ng outer layer ng coat. So, each Clathrin subunit is nagko-consist sila ng mga 3 large and 3 small polypeptide chains na kung saan nagbubuo yung 3 large at 3 small polypeptide chains ng 3-leg structure, which is sinatawag natin na triskelion or triskelion. So, yung Clathrin triskelion naman, nag-assemble ito sa nag-assemble siya into a basketball framework of hexagons or pentagons to form coated pits or yung buds na tinatawag natin sa cytosolic surface ng mga membranes. Kaya si Clathrin triskelion is siya yung nagde-determine ng geometry ng Clathrin cage. So next, COP1 coated vesicles. So bud from Golgi compartments, a protein complex that coats vesicle transporting proteins from the cis end of Golgi complex back to the rough ER. So, si COP1, isa siyang protein complex na kung saan nagko-coat siya ng vesicle, nagtatransport siya ng mga proteins galing sa cis end ng Golgi complex pabalik sa rough endoplasmic reticulum. Well, si COP2 coated vesicles naman, ang role naman niya is nagbabad siya galing sa endoplasmic reticulum and then nagpo-form siya. Yung COP2 is formation yun ng mga membrane vesicles na kung saan nag-export sila ng mga newly synthesized proteins na galing sa ER or endoplasmic reticulum. So, ngayon, meron tayong table ng 3 types of protein-coated vesicles. Clathrin-coated vesicles mediate transport from the plasma membrane to early endosomes, immature secretory vesicles to Golgi apparatus to late endosomes. COP1-coated vesicles naman mediate transport from early Golgi back to ER, Golgi apparatus to plasma membrane, and between various Golgi cisternae. While sa COP2 coated vesicles naman is nagme-mediation ng transport from ER to the Golgi apparatus. So, yung mga nasa baba, ayan yung mga picture nung nilang tatlo. So, next, adapter proteins select cargo into Clathrin-coated vesicles. So, yung adapter proteins, another major coat components sila ng Clathrin-coated vesicles na kung saan bumubuo sila ng discrete inner layer ng coat, which is, nakaposition siya between the Clathrin cage and the membrane. So, they bind the Clathrin coat to the membrane and trap various transmembrane proteins or receptors na nagka-capture ng mga soluble cargo molecules inside the vesicle na tinatawag nating cargo receptors. So, itong si cargo receptors, siya yung ayan yung mga soluble cargo molecules na nasa loob ng vesicle. So, in this way, yung mga adapter proteins, namimili sila ng specific set ng transmembrane proteins together with soluble proteins na nakikipag-interact sa kanila. And then, pinapackage sila into each newly formed Clathrin-coated transport vesicle. So, adapter proteins found in other coats also bind to phosphoinositides. So, yung adapter proteins daw na nakikita sa ibang coat naman is nagbabind din sila sa mga phosphoinositides, which not only have a major role in directing when and where coats assemble in cells. So, si phosphoinositides, hindi lang siya merong major role, which is yung nagdadirect siya ng mga kung saan yung mga coats is nag-assemble sa cell. But also, si phosphoinositides is ginagamit din sila much more widely as molecular markers. Ginagamit sila ng mga molecular markers ng compartment identity. So, this helps. So, once na meron tayong phosphoinositides na nagsaserve ng mga molecular markers, so matutulungan nito yung makontrol yung vesicular traffic. So, para mabalansin naman natin yung vesicle traffic sa isang compartment, so yung mga cold proteins is kailangan ma-assemble lang sila when and where they are needed. Kung kailan lang sila, kailangan doon lang sila mag-assemble. Additional, yung mga local production ng PIP postpoinositases, nagkakaroon sila ng major role or major part in regulating the assembly of Clathrin coats sa nasa plasma membrane and then nasa Golgi apparatus. So, Rab proteins nag-guide the transport vesicles. So, to ensure an orderly flow of vesicle traffic, para ma-ensure natin yung maayos na flow ng vesicle traffic, kailangan namang yung mga transport vesicles is highly accurate in recognizing the correct target membrane with which to fuse. So, kung saan lang talaga yung appropriate target membrane nila is doon lang talaga sila mag-fuse. So, yun nga, dahil sa diversity and crowding na nangyayari sa mga membrane system sa cytoplasm, so yung mga vesicle, mas nagkakaroon sila ng, parang mas na-encounter nila yung mga many potential target membranes before nila mahanap yung correct target membrane na pag-fuse nila. So, this crucial process occurs in two steps. So, yung Rab proteins, which is yung Rab proteins and then yung Rab effectors na nagdadirect sa vesicle para maano nila yung mga specific spots on the correct target membrane. So, second, snare proteins and regulators, sila naman yung nagme-mediate ng fusion sa lipid bilayer. So, itong si Rab proteins, si Rab effectors, snare proteins, and regulators is mas madidiscuss mo natin sa next slides. Rab proteins, nagpe-play siya as a central part in the specificity of vesicle transport. So, each Rab protein is meron silang kalakip na mga one or more membrane-enclosed organelles in the secretory or the endocytic pathways. And each of these organelles na nakapadoob dun sa Rab protein is meron silang at least one Rab protein on its cytosolic surface. And then si Rab proteins naman, kino-consider siya as ideal molecular markers, which is na-identify nito yung each membrane type and then nagka-guide nito yung vesicle traffic na nangyayari between them. So, yung Rab proteins naman can function on transport vesicles and on target membranes, or pwedeng both transport vesicles and then both target membranes. So, next, Rab effectors. Si Rab proteins naman is nagbabayin siya sa other proteins, which is sinatawag natin na Rab effector. So, Rab effectors, tandaan natin, ito yung other proteins na kung saan dito nagbabind si Rab proteins. So, si Rab effector naman, kino-consider siya as downstream mediator ng vesicle transport, ng membrane tethering, and then ng membrane fusion. So, some Rab effectors are motor proteins, which is yung motor proteins naman, nagpo-propel sila ng vesicles along acting filaments or microtubules to their target membrane. While yung ibang Rab effectors naman is tinatawag natin tethering proteins, na kung saan sila naman yung nagsaserve ng mga fishing lines, nagsaserve as fishing lines that can extend to link two membranes more than 200 meter apart. And then si Rab effectors naman, pwede siya makipag-interact kay snares to couple membrane tethering to fusion. Additional kay tethering proteins, so bukod sa kaya nitong mag-link ng two membranes more than 200 meter apart, so si tethering protein nakikipag-interact din siya sa mga wide variety ng mga ibang proteins na kung saan nagpa-facilitate ito ng membrane fusion step. So, let's proceed to snares mediate membrane fusion. So, once na yung transport vesicles is na-tethered na siya dun sa target membrane niya, di ba ang mangyayari is i-unload na niya yung cargo na daladala niya by means of membrane fusion. So, si membrane fusion, nagre-require siya ng pagdala ng lipid bilayers ng dalawang membranes sa isa't isa para mag-merge silang dalawa. So, si snare proteins, also called snare for short, sila naman yung nagka-catalyze ng membrane fusion reactions in vesicle transport. So, yun nga, nangyayari yung membrane fusion kapag yung transport vesicle is na-tethered siya sa sa target membrane niya, then nag-unload na siya ng kanyang cargo. So these transmembrane proteins exist as complementary beds. So with Bisners, si Bisners is makikita siya sa vesicle membrane, and then si Tisners is makikita naman sa target membrane. Si Bisner, ano lang siya, single polypeptide chain, whereas si Tisner naman is nagko-compose siya ng tatlong proteins. Okay, so si Tisner saka si Bisner, meron sila parehas ng mga helical domains. Okay, so yung helical domains ng isa, nagra-wrap siya doon, or bumababalot siya doon sa helical domains ng isa pa in order to form a very stable helix bundle. So, di ba, kunwari, yung helical domain ni Tisner is bumabalot siya doon sa helical domain ni Bisner para magkaroon or mabuo yung very stable helix bundle. Additional Sisners, nagpo-provide siya ng additional layer ng specificity sa transport process, which is, ginagawa niyo yon para ma-ensure na yung mga vesicles is nag-fuse only with the correct target membrane. So, in the cell naman, other proteins is nare-recruit sila sa fusion site, which is yung mga Rabe vectors, nakikipag-cooperate sila sa snares para ma-accelerate yung fusion. And then yung fusion na nangyayari, hindi siya palaging nakasunod sa after ng mga V-snares and then ng mga T-snares ng mga pair. For vesicle transport naman to operate normally, yung mga transport vesicles must incorporate the appropriate snare and then the appropriate Rab proteins. So now, let's proceed na sa summary ng molecular mechanism of membrane transport. So there are various types of coated vesicles. Cyclotin coated vesicles na nagme-mediate na transport from plasma membrane and trans-Golgi network. COP1 coated vesicles naman mediate from Golgi apparatus to plasma membrane, and COP2 coated vesicles mediate transport from ER or endoplasmic reticulum to Golgi apparatus. So, yung coats, meron silang two-layered structure, which is yung inner layer na binubuo ng mga adapter proteins, and then nagli-link siya sa outer layer or yung cage na tinatawag sa vesicle membrane. Then, sabi dyan, the coat is shed before the vesicle fuses within its appropriate target membrane. So, nagsished muna yung coat bago siya mag-fuse sa appropriate target membrane niya. So, post-poinositase naman, ginagamit sila as molecular markers to compartment identity, which is, nag-help to control the vesicular traffic. So, to balance the vesicular traffic from a compartment, yung coat proteins kailangan, i-assemble lang sila kung kailan sila kailangan. And then, to ensure naman the orderly flow of vesicle traffic, yung transport vesicle must be highly accurate in recognizing the correct target membrane or the appropriate membrane kung saan na sila mag-fuse. Then, si Rab effectors, sabi dyan, serve as downstream mediators of vesicle transport. And then, some Rab effectors are motor proteins, which is nagpo-propel ng vesicles. And then, yung tethering proteins, which is nagsaserve as fishing lines. So, Bisners, ayan, vesicle membranes. Tisners is target membranes. Additional, so yung interior space or yung lumen na tawag natin, ng mga membrane enclosed compartment is equivalent siya sa lumen ng mga other membranes enclosed compartment and cell exterior. So through exocytosis naman, yung secretory pathway is dinedeliver niya yung newly synthesized proteins either sa plasma membrane or sa extracellular space. While si endocytosis naman is yung mga cells, nire-remove niya yung mga plasma membrane compartments and dinideliver ito sa mga internal compartments na tinatawag natin na endosomes. So next, let's proceed to transport from the ER through Golgi apparatus. So yung transport from one compartment to the next, syempre nag-involve yun ng delicate balance between forward and backward or yung mga retrieval transport pathways. So, yung mga ibang transport vesicles daw is namimili sila or nagsaselect sila ng mga cargo molecules and then nililipat nila ito sa next compartment in the pathway. While yung mga ibang transport vesicles naman is nagre-retrieve sila ng mga escape proteins and then binabalik nila ito sa previous compartment na kung saan dun nagno-normally function yung mga yung mga escape proteins. And then sabi dyan, Golgi apparatus, also called Golgi complex, is a major site of carbohydrate synthesis, as well as sorting and dispatching station for the products of endoplasmic reticulum. So yung Golgi apparatus naglalay siya sa sa exit route na galing sa ER and then meron siyang mga large proportion ng carbohydrates na kung saan naka-attach yun sa oligosaccharide side chains to many proteins and lipids that the ER sends to it. So a subset of these oligosaccharide groups is nagsuserve siya as a tag. So yung tag na yun is nagdadirect sila ng mga specific proteins into vesicles and then tinatransport nila to sa lysosome. Only proteins that are properly folded and assembled can leave the ER or endoplasmic reticulum. So to exit from the ER, syempre yung mga proteins daw para makalabas sila ng galing sa ER is kailangan sila properly folded. If they are subunits of multi-protein complexes, they need to be completely assembled naman. So those that are misfolded or incompletely assembled transiently is remained in the ER. So meaning na hindi sila makaka-exit. So, kung saan kapag halimbawang merong mga misfolded or mga incomplete assembled, ang mangyayari dyan is binabone sila to chaperone proteins. Wherein, yung mga chaperone proteins naman, sila naman yung nagko-cover up ng mga exit signals or somehow anchor the proteins in the ER. So, after yung mga transport vesicles is mabaded na galing sa mga ER exit sites is ang mangyayari dyan, magsha-shed ulit sila ng coat nila and then after nila mag-shed ng coat nila, doon na sila ulit magsisimula mag-fuse with one another. The fusion of membranes from the same compartment is called homotypic fusion. A membrane from one compartment fuses with the membrane of a different compartment is called heterotypic fusion. So, tatandaan nyo guys na kapag sinabi natin homotypic fusion is, yung isang membrane is nagpo-fuse siya sa same compartment lang. Hindi siya nagtatransfer sa isang compartment. Meaning na doon lang talaga siya nagpo-fuse. So, kapag sinabi namang heterotypic fusion, yung membrane na galing sa isang compartment is nagpo-fuse siya sa different compartment. So, meaning na nagtatransfer pa siya sa ibang compartment para mag-fuse, which is tinatawag natin yung heterotypic fusion. So, the structures formed when ER-derived vesicles fuse with one another are called vesicular tubular clusters. So, yung mga nabubuo ng structure kapag yung mga ER-derived vesicles is nagpo-fuse with one another is tinatawag sila na mga vesicular tubular clusters. So, nag-generate sila continually and nagpa-function sila as transport containers, which is nagbe-bring sila ng material galing sa ER papunta sa Golgi apparatus. During their passage through the Golgi apparatus, yung mga transported molecules is nag-undergo sila ng mga ordered series ng covalent modification, which is yung mga each Golgi stack, meron silang two distinct phases na nangyayari. So, sa cis phase, ayan yung entry phase, and then yung trans phase, ayan naman yung exit phase. So, each composed of a network of interconnected tubular and cisternal structure, which is yung cis-Golgi network or CGN, and then yung trans-Golgi network, which is yung TGN. So, tandaan natin na si cis phase, entry phase, so si trans phase, exit phase. Then, meron silang dalawang network, which is si cis-Golgi network, and then si trans-Golgi network. So, si cis-Golgi network, collection siya ng mga fused vesicular clusters, which is papunta yon sa galing siya sa ER. So, yung mga proteins and lipids daw is nag-enter sila sa cis-Golgi network and then nag-exit sila sa trans-Golgi network. Meaning na si cis-Golgi network, dyan papasok si cis-phase and then si trans-Golgi network is dyan papasok si trans-phase. Yung both networks, which is yung cis-Golgi network and then yung trans-Golgi network is napaka-important nun sa protein sorting. So, yung proteins na nag-enter sa cis-Golgi network can move in Golgi apparatus or pwede silang bumalik sa ER. While yung mga proteins naman na nag-exist sa trans-Golgi network is sinusort pa sila according to their next destination or pwede silang bumalik sa earlier compartment nila. So for the summary, Golgi apparatus is a major site of carbohydrate synthesis. Also, it is a sorting and dispatching station for the products of the ER. So, si oligosaccharide groups naman, nagsiserve naman siya as a tags to direct specific proteins into vesicles na nagtatransport into lysosomes. So, sabi dyan, to exit from the ER, yung mga proteins daw is kailangan properly folded sila. So, after ng mga transfer vesicles na magbad galing sa mga ER exit sites, and after nila mag-shed ng kanilang mga coat, doon sila nagsisimula mag-fuse sa isa't isa. So, si homotypic fusion naman, ito yung mga membrane fuse from the same compartment. And then si heterotypic fusion, ito yung isang membrane nagpo-fuse siya sa different compartment. So, vesicular tubular clusters function as transport containers, which is nagbibring siya ng material galing sa ER to papuntang Golgi apparatus. So, Golgi stack naman, meron siyang two distinct phases, which is yung cis phase or yung entry phase, and then yung trans phase or yung exit phase. And then, meron din nagkukompose nito ng mga network ng cisternal structures, which is yung cis-Golgi network and then yung trans-Golgi network. So, cis-Golgi network, collection siya ng mga fused vesicular tubular clusters, and then sabi dyan, yung mga proteins and lipids nag-enter sila sa cis-Golgi network and then nag-exit sa trans-Golgi network. Transport from trans-Golgi network to lysosomes. So, lysosomes are membrane-enclosed organelles filled with soluble hydrolytic enzymes that digest macromolecules. So, example ng mga hydrolytic enzymes is yung mga proteases, nucleases, glycosidades, lipases, phospholipases, phosphatases, and sulfatases. So, all are acidic hydrolysis. So, yung mga binanggit ko kanina is lahat yun are acidic hydrolysis na kung saan nagwo-works best sila sa kanilang mga acidic pH. So, sa lysosome, nagpo-provide ng acidity, maintaining an interior pH of about 4.5 to 5.0. So, transporting proteins in the lysosome membrane is, ang nangyayari doon, kinikarry nila yung final products ng mga digestion ng macromolecules such as yung mga amino acids, yung mga sugars, yung mga nucleotides papunta sa cytosol wherein yung mga cell can either reuse or secrete them. So, yung mga lysosomes, madalas sila nakikita sa mga eukaryotic cells, sa lahat ng eukaryotic cells. So, yung lysosome is na-discovered siya through biochemical fractionation of cell extract. And then later on, saka lang siya nakita sa electron microscope. So next, sinasabi dyan na multiple pathways deliver materials to lysosomes. Lysosomes, isa sa mga role ng lysosome is ito yung nagsaserve as meeting places where several streams of intracellular traffic is nagko-converge sila. So a route that leads outward from the ER via Golgi apparatus delivers most of the lysosomes digestive enzyme. So yung ruta daw na ER via Golgi apparatus, dyan yung yung ruta na kung saan dinedeliver mostly yung mga lysosomes, digestive enzymes. So, meron tayong 4 paths from different sources that feed substances into lysosomes for digestion. So, yan. This degradation paths is the first followed by macromolecules. So, yung first degradation paths is sinusundan siya ng macromolecules taken up from extracellular fluid by endocytosis. So, first is endocytosis. Second, similar pathway, meron tayong yung which is nakikitulong siya sa mga phagocytic cells such as yung mga macrophages. Then, and is dedicated to engulfment or phagocytosis of microorganism to form phagosomes. So, once na nagkaroon ng engulfment or ng phagocytosis tinatawag, dito na nabubuo yung mga phagosomes. So, second, phagocytosis. Then, third pathway natin is macropinocytosis. Sa macropinocytosis naman, ang sinespecialize naman ito is yung mga non-specific uptake ng mga fluids and then ng mga membranes, which is na ito yung yung mga fluids sa membranes na naka-attach sa plasma membrane. And then yung fourth pathway natin, sinatawag natin autophagy. Si autophagy naman, nag-originate naman siya sa cytoplasm, and then ang role nito is to digest cytosol and worn out organelles. So, all cell types, nagdi-dispose sila ng mga obsolete parts by a lysosome-dependent process which tinatawag natin na autophagy or self-eating. Autophagy can either be selective or non-selective. So, non-selective autophagy, yung bulk portion ng cytoplasm is nasa-sequestered sa autophagosomes. While sa selective autophagy naman, yung specific cargo is nakapackage siya into autophagosomes, which is nagko-condense siya ng little cytosol, and yung mga shape nito is nagre-reflect din siya shape ng cargo. So, additional, yung non-selective autophagy, nag-occur din siya sa mga starvation conditions na kung saan yung mga, once na nag-limit na yung mga external nutrients, and then, yung mga metabolites na nanggaling sa digestion ng cytosol is pwede yung makatulong para mag-survive yung isang cell. While sa selective autophagy naman is used to destroy invading microbes. So, additional din, yung tinatawag natin na mitophagy, ayun naman yung selective autophagy na kung saan selective autophagy siya ng mga worn out or mga damaged mitochondria. Transmembrane M6P receptor proteins naman, which is present sa TGN or Trans-Golgi network, is sila naman yung nagre-recognize sa mga map groups, and then nagbabind sila sa mga lysosomal hydrolysis sa luminal side ng mga membrane, and then sa mga adapter proteins na kung saan nag-assemble ng mga cluttering coat sa cytosolic side. And then yung mga receptors, tinutulong din sila mag-package ng mga hydrolysis into cluttering coated vesicles na kung saan itong mga vesicles ito is nagbabad siya from Trans-Golgi network, and then dinideliver nila yung mga contents ito sa early endosomes. So, not all hydrolyzed molecules that are tagged with M6P is napupunta sa lysosomes. Okay, so yung mga iba is nag-escape sila sa normal packaging process ng Trans-Golgi network, and then natatransport sila sa default ng cell surface na kung saan doon na din sila sinesecrete into extracellular fluid. Then yung mga iba naman, ang nangyayari is nagkakaroon ng detour sa plasma membrane na kung saan doon sila nire-recapture, doon nagkakaroon ng mga recapture ng mga escape lysosomal hydrolysis, and then binabalik sila sa lysosomes. So, yung mga M6P groups, kailangan mas mai-add sila or must be added only to the appropriate glycoproteins in the Golgi apparatus. Okay, so yung mga targeting ng mga material sa lysosomes is hindi pa to yung tapos ng pathway. So, yung mga lysosomal secretion ng mga undigested debris content or na mga undigested content is nag-enable sila to eliminate the indigestible debris. So, yung mga ibang cell types, however, is nagko-contain daw sila ng mga specialized lysosomes, which is parang na-accord nila yung necessary machinery for fusion with the plasma membrane. Then, under certain conditions naman, yung mga multivesicular bodies natin is nagpo-fuse sila sa pwede din sila mag-fuse sa plasma membrane. So, kapag nangyari yon, yung mga intraluminal vesicles is magre-release sila sa mga cells. And then, yung mga circulating small vesicles naman, tinatawag naman natin sila mga exosomes. So, yung mga exosomes na yun is mostly nakikita natin yun sa mga blood. And then, ang nangyayari is yung mga exosomes is ang role nila is to transport components between cells. So, some exosomes may derive from direct vesicle budding events at plasma membrane. So, summary, lysosomes are specialized for the intracellular digestion of macromolecules. So, yun na. Yung lysosomes, nagko-contain sila ng mga soluble hydrolytic enzymes that operates best at pH 5, which is yung internal pH ng lysosomes. So, lysosomes are found in all eukaryotic cells. Lysosomes are meeting places where several streams of intracellular traffic converge. Okay? So, yung lysosomes nagsaserve siya as meeting places sa mga several streams ng intracellular traffic. Then, transporting proteins in lysosomes is once na nag-transport ng mga proteins, is dinadala nila yung mga final product ng mga digestion of macromolecules such as yung amino acids. And then, meron tayong 4 paths na kung saan nag-feed ng substances into lysosomes for degradation. So, yan. Number 1, macromolecules taken up from extracellular fluid by endocytosis. Number 2, macrophages is dedicated to engulfment or phagocytosis. Then number 3, macropinocytosis. Ito naman yung mga specific uptake ng fluids na naka-attach sa plasma membrane. Number 4, autophagy used to digest cytosol. Then, dito na papasok sila non-selective autophagy sa kasi selective autophagy. Si non-selective autophagy, mag-occur siya sa mga starvation conditions, while si selective autophagy is ginagamit siya para ma-destroy yung mga invading microbes. Then si mitophagy naman, ito naman yung selective autophagy ng damaged mitochondria. So, transmembrane M6P receptors, dito naman tinutulungan naman ito na i-package yung mga hydrolyses, then magiging Clathrin-coated vesicles sila. And then once naging vesicles na, i-deliver na nila yung content sa early endosomes. So, exosomes naman, ito naman yung mga small vesicles used to transport components between cells. Next, proceed na tayo sa transport into the cell from the plasma membrane or yung tinatawag natin na endocytosis. So, sabi dito, the routes that lead inward from the cell surface start with the process of endocytosis. Yung mga ruta na papunta sa mga cell surface is mag-start sila sa process ng endocytosis na kung saan yung mga cells is nagtitake sila ng mga plasma membrane components, fluid solutes, macromolecules, and substances. So, yung mga endocytose cargo, nakapaloob dito yung mga extracellular matrix components. Example na lamang dito is yung mga cell debris, bacteria, viruses, and even other cells. So, through endocytosis din, yung cell, nagre-regulate siya sa composition ng kanyang plasma membrane in response to changing extracellular condition. So, in endocytosis, the material to be ingested, yung material na i-ingest or kailangan ma-ingest is nakapaloob siya sa small portion ng plasma membrane and then pinipinch off siya to form endocytic vesicle na nagko-contain yun ng ingested material. Okay, so most eukaryotic cells is constantly nagpo-form sila ng mga endocytic vesicles, which is a process called pinocytosis or cell drinking. And then, yung mga iba namang specialized cells na nagko-contain ng mga dedicated pathways, is nagtitake sila ng mga large particles, which is a process naman called phagocytosis or cell eating. And then, sa once na nag-generate na yun sa mga plasma membrane, yung mga mostly, yung mga endocytic vesicles daw is nagpupuse sila sa mga receiving compartment. So, which is yung the early endosomes where kung saan yung doon sa early endosomes na yun is yung mga internalized cargo is sinasorted na sila. So, late endosomes, form from a bulbous vacuolar portion of endosomes, which is tinatawag natin na process called endosome maturation. Okay? So, as an endosome mature, kapag halimbawang nag-mature na isang endosome, it ceases recycled material to plasma membrane. Okay? And then, nangyayari, yung mga remaining contents niyan is mapupunta sa degradation. Then, yung mga late endosomes is nagpupuse sila with one another and with lysosomes. Then, kaya sila nagpupuse, yung mga late endosomes, nagpupuse sila with one another and then nagpupuse sila sa lysosome to form endolysosomes, which degrade their contents. So, virtually, yung mga karamihan na mga eukaryotic cells is, nag-i-ingest sila ng portions of their plasma membrane in the form of small pinocytic core, tinatawag natin na endocytic vesicles. So, the rate at which plasma membrane is internalized in this process of pinocytosis varies between cell types. So, since a cell surface area and volume is remain unchanged during this process, during yung process ng pinocytosis, diba hindi nagbabago yung cell surface area and then yung volume, it is clear that the same amount of membrane being removed by endocytosis is being added to the cell surface by the converse process of exocytosis. So, si endocytosis, siya yung nagre-remove. Okay? So, si exocytosis naman, yung converse process naman ni exocytosis is, siya naman yung nag-a-add sa cell surface. So, in this sense, endocytosis and exocytosis are linked processes that can be considered to constitute an endocytic exocytic cycle. Okay, so si endocytosis and then si exocytosis is palagi silang mag-match or palagi silang magkadikit, which is kaya sila tinatawag na link processes. So yung endocytic part naman doon ng cycle often begins at glatrin-coated pits, which is ang nangyayari dito is ini-invaginate ito into the cell and then pinipinch off ulit ito to form a glatrin-coated vesicle naman. So, yung mga early endosomes have tubular and vacuolar domains. Okay, so mostly yung mga membrane surface is in the tubules and then mostly mga volume naman ng mga membrane surface is in the vacuolar domain. Okay, so membrane surface, okay, kita natin sa tubules and then yung mga mostly yung mga volume nito is nasa vacuolar domain. So, during endosome maturation, yung dalawang domain, meron daw silang different fates, okay? Sabi dyan, the vacuolar portion of the early endosomes are retained and transformed into late endosomes and the tubular portions shrink. So, yung fate nila is yun nga, yung vacuolar portions ng early endosomes are retained and then transform sila into late endosomes and the other one is the tubular portions is nag-shrink. So, phagocytosis is a special form of endocytosis in which a cell uses large endocytic vesicle which is tinatawag natin na phagosome. Ang nangyayari dito is to ingest a large particle such as microorganisms and then yung dead cells. So, phagocytosis isa siyang cargo triggered process, which is kailangan itong mag-require ng activation ng mga cell surface receptors na kung saan ang goal niya is to transmit signals to cell ends. So summary, cells ingest fluids, molecules, and particles by endocytosis in which localized regions of plasma membrane invaginate and pinipinch off sila para mag-form ng endocytic vesicles. Okay? So the cells remain the same size because most of the plasma membrane components that are endocytosed are continually returned to the cell surface by the means of exocytosis. So it is clear that the same amount of membrane being removed by endocytosis is being added to the cell surface by converse process of exocytosis. So, malinaw na sinasabi dyan na yung same amount ng membrane na ni-remove ng endocytosis is ayun din yung same amount ng membrane na ina-add ng exocytosis, ng converse process ng exocytosis sa cell surface. So endocytosis and exocytosis are linked processes that can be considered to constitute or to form an endocytic and exocytic cycle. So endocytic part of cycle begins at clathrin-coated pits, which is nag-i-invaginate sila into the cell, and then after nun, pinipinch off sila to form clathrin-coated vesicle. While si phagocytosis naman is a special form siya ng endocytosis na kung saan yung mga cell, gumagamit sila ng mga large endocytic vesicles na tinatawag natin ng mga phagosomes in order to ingest large particles such as yung mga microorganisms. And then si phagocytosis is a cargo-triggered process that requires the activation of the cell surface receptors. So, the best characterized trigger na nakakapag-trigger talaga sa phagocytosis is yung antibodies natin. Last but not the least, we have the transport from trans-Golgi network or yung TGN to the cell exterior na tinatawag nating exocytosis. So, yung mga transport vesicles na nakadestino sa plasma membrane is normal na sa kanila na mag-live sa trans-Golgi network in a steady stream as irregularly shaped tubules. So, yung mga membrane proteins and then yung mga lipids in these vesicles is nagpo-provide sila ng mga new components for the cell's plasma membrane. While yung mga soluble proteins naman inside the vesicles na to is sinisecrete sila papunta sa extracellular space. So, the fusion of the vesicles with plasma membrane is called exocytosis. Exocytosis, yung mga lahat ng cells is nire-require tong constitutive secretory pathway which is nagkikipag-cooperate siya continuously. So, specialized secretory cells, meron silang mga second secretory pathway in which yung mga soluble proteins and then yung mga other substances are initially stored in secretory vesicles, which is later on nire-release sila through exocytosis. So, yung mga cells that are specialized for secreting some of their products rapidly on-demand concentrate and store these products in secretory vesicles. Secretory vesicles form from the TGN, nabubuo sila sa TGN and then later on, nire-release sila yung content sa cell exterior by the means of exocytosis in response to specific signals. So, yung mga secretory and then yung mga membrane proteins, nagiging concentrated sila as they move from the ER through the Golgi apparatus because of the extensive retrograde retrieval process na minemediate ng COP1-coated transport vesicles. And then, dahil doon, yung mga COP1-coated na yun is nagkikarry sila ng mga soluble ER resident proteins back to the ER while excluding the secretory and membrane proteins. So, membrane recycling is important for returning Golgi components to the Golgi apparatus. So, yung membrane recycling, tandaan natin na ito yung yung binabalik yung mga Golgi components sa Golgi apparatus as well as yung mga concentrating the contents of secretory vesicles. So, because the final mature, yung kapag nangyari na yung final mature, ang nangyari is yung mga secretory vesicles are so densely filled with contents. So, yung mga cells, pwede sila mag-disgorge ng mga large amount ng material promptly by the exocytosis when triggered to do so. So once na napuno na yung secretory vesicle, kailangan na niyang ma-reach yung site ng secretion, which in some cells is far away from transcolchi network. So yung mga motor proteins naman, ito naman yung mga nagpo-propel ng vesicles along axonal microtubules whose uniform orientation guides the vesicle in proper direction. So yung motor proteins, ang nangyayari dyan, yung uniform orientation is ginaguide niya yung mga vesicles in proper direction, and then pinopropel nito yung mga vesicles na mga nasa axonal microtubules. Then sa microtubules naman, ang nangyayari naman dito is ginaguide naman nila yung mga transport vesicles papunta sa cell surface para sa constitutive exocytosis. Sa constitutive exocytosis naman, dito naman nangyayari yung kung saan yung mga transport vesicles nagko-contain sila ng mga materials para sa constitutive release na magpo-fuse sa plasma membrane. So once na naka-arrive na sila doon, yung mga secretory vesicles in the regulated pathway wait at the membrane, okay? Nag-aantay sila until yung mga cells naka-receive na sila ng signal na pwede na sila mag-secrete, and then pwede na sila mag-fuse. So meaning na hindi sila pwedeng mag-secrete, and hindi sila pwedeng mag-fuse kapag hindi pa sila nakaka-receive ng appropriate signal para doon. And then yung signal na yon can be an electrical nerve impulse or yung action potential, or it can be an extracellular signal molecule such as yung hormone. Then kapag naman yung secretory vesicle is nag-fuse siya sa plasma membrane, yung mga content nito is nadi-discharge from the cell by the means of exocytosis. And then ang mangyayari doon, yung mga membrane nila is magiging part na siya ng plasma membrane. So after ng removal nila galing sa plasma membrane, ang mangyayari sa yung mga proteins ng mga secretory vesicle membrane are either mare-recycled or masya-shuttled to lysosomes for degradation. So next, control of membrane traffic has a major role in maintaining the composition of the various membranes of the cell. So sa pag-control nga ng membrane traffic, ang pinaka-major role niya is to maintain the composition of the various membranes of the cell. Okay, so to maintain each membrane and closed compartment in the secretory and endocytic pathway at a constant size, the balance between the outward and the inward focus of membrane needs to be precisely regulated. So para ma-maintain natin yung mga membrane and closed compartment ng mga secretory sa ng mga endocytic pathway at a constant size is kailangan nating mabalance yung mga outward saka yung mga inward focus ng mga membrane, which is kailangan nating ma-precisely regulated. So, in order for the cells to grow naman, the forward flow needs to be greater than the retrograde flow. So that yung mga membrane can increase in an area. So, tandaan natin, para naman mag-grow yung mga cells, yung forward flow needs yun na mas malaki siya kumpara sa retrograde flow. And then, for cells naman to maintain at its constant size naman, the forward and the retrograde flows must be equal. Okay? Kapag naman yung mga cells, para ma-maintain nila yung mga constant size nila, kailangan yung mga forward and then yung mga retrograde flows is equal lang, meaning na balance na silang dalawa. So summary for the exocytosis, the fusion of the vesicles with the plasma membrane is tinatawag na natin na exocytosis. So cells can secrete molecules by exocytosis in either a constitutive or regulated fashion. Whereas yung mga regulated pathways is nag-ooperate lamang sila sa mga specialized secretory cells. While yung constitutive secretory pathway is nag-ooperate naman sila sa lahat ng eukaryotic cells, which is characterized by continual vesicle transport from TGN to papuntang plasma membrane. So in regulated pathways, the molecules are stored either in secretory vesicles or in synaptic vesicles, which is, ito sa regulated pathways nga, yung mga secretory vesicles is hindi sila nag-fuse sa mga plasma membrane to reduce their contents until hindi sila nakakatanggap ng appropriate signal para dun. So when the secretory vesicle fuses with the plasma membrane, di ba, yung mga content nito is nadi-discharge na from the cell by the means of the exocytosis. And then therefore, yung mga membrane nila is magiging part na yun ng plasma membrane. So, yung mga secretory proteins naman is nagiging concentrated sila kapag naman nagkakaroon ng formation and then ng maturation sa mga secretory vesicles. Okay, secretory vesicles is nagko-contain sila ng mga proteins for secretion bud from the transgolgi network. And then yung mga synaptic vesicles naman, which are confined to nerve cells and some endocrine cells, formed from both endocytic vesicles and endosomes. So synaptic vesicles is nabubuo sila from both endocytic vesicles and then endosomes. And then, si synaptic vesicles is siya naman din yung nagmemediate ng mga regulated secretion of small molecules, neurotransmitter at axon terminal of nerve cells. Proceed to our research article, which is probing and manipulating intracellular membrane traffic by microinjection of artificial vesicles. So, in this study, yung mga researchers, gumamit sila ng extracellular delivery of engineered membranes with the goal to assess whether yung mga artificial vesicles or yung mga nanoparticles can be created in test tube. Yung meaning na yung mga artificial vesicles are functional daw in highly complex cellular process of membrane trafficking and which can be used to sense the intracellular environment and to modulate intracellular function. So, ang pinaka-goal dito is to assess whether yung mga artificial vesicles can be created in test tube, meaning na kung makikita ba sa test tube yung visible, yung pagiging visible ng mga artificial vesicles. So, furthermore, by incorporating appropriate sensors or affinity tags into LUVs or large unicellular vesicles, it is possible to detect pH and to change the dynamics of endosomal trafficking. Is this mga goal nila dito is tinitingnan nila kung yung mga appropriate sensors pa daw or yung mga affinity tags sa mga LUVs or yung mga large unicellular vesicles is possible na ma-detect nila yung mga pH and possible na mabago nila yung dynamics ng endosomal trafficking. So yung results, microinjection of nanoparticles does not affect cell viability. Meaning na yung mga results ito, nag-indicate na yung microinjection protocol does not have any negative impact on cell viability or organelle distribution. Meaning negative impact siya sa cell viability. So, sa LUVs naman, LUVs are transported along microtubules via dynein or dynection complexes but only with considerable delay after microinjection. So, taken together, nag-undergo din ng sinasabi ng mga researchers na yung injected SUVs na ginamit nila is nag-undergo ng random diffusion and are quickly captured by the Golgi complex with no involvement of cytoskeleton-based transport. In contrast, yung mga injected LUVs na ginamit nila is nagsishow siya ng reduced diffusion. So, snare proteins suffice for specific targeting of liposomes to endosomes. Sabi nila yung particular daw yung mga endosomal system is nagko-contain sila ng multiple trafficking routes na connected sa TGN with plasma membrane and degradative link of pathway with identity and targeting shuttle vesicles that is being governed by zip code proteins such as yung mga RABs, yung mga snare, and yung mga phosphoinositase. Nasabi din nila na yung mga only integral membrane constituents that are known to be essential for all trafficking organelles of the secretory pathways, nag-include sila ng mga snare proteins, hindi lamang para makatalyze yung fusion, but also participate in the recruitment of the specific tethering factors. So here are my resources and references, and thank you for listening.